that, I am excited to begin session 2, Models of Cerebral Creatine Deficiencies. Our first speaker for this session is Dr. Laura Baroncelli. Dr. Baroncelli is a researcher at the Institute of Neuroscience at CNR in Pisa, Italy, and is also affiliated with the Stella Maris Foundation. Her research focuses on neurodevelopmental disorders, rare disease, and epilepsy. As you know, she's heavily involved in functional brain imaging. Her group initiated the FNIRS work, and she is now doing this work in collaboration with Dr. Michela Faglioni at the Boston Children's Hospital. She will be speaking about models of creatine transporter deficiency today. Can use this? Okay. Test it and make sure it works.
Yes. And come back. Okay. So good morning, everyone. First of all, I would like to thank the organizers for inviting me here and putting together year after year this amazing symposium. It is always a pleasure for me to be here and meet all of you. And I'll also use the floor to tell you that, as Sanjita just mentioned, we are doing FNIRS recordings in the other building, the buzzer room, I guess. And we have some free spot for the afternoon. So if some families want to step by, take a look, and whatever, please feel free to reach me. And of course, thanks to all the families already enrolled on the studies. So today, my goal is to take a step back from my own research and provide you an overview of the preclinical models of creatine transporter deficiency, in particular of the animal models. But first, before we start, I would like to acknowledge the work of the members of my team, because even if today I'm not presenting to you our unpublished results, I feel that my perspective always emerges from the discussion with all of them. So I would like to thank past and present members of my team, because they are very talented researchers. So let me start with the very first fundamental question, which is, why do we need animal models in the first place? Well, as you know, we can learn a lot from the study of patients. We can describe very well the phenotype of the disorders. We can also understand how the disorder manifests across different ages, thanks to the different natural history studies that they have taken over the last years. But there are some limits to what we can understand just from studying patients alone. And this is where animal models become essential, because in animal models, we can study cellular and molecular mechanisms, vulnerable brain circuits. We can basically study causative disease mechanism in a controlled way. And we can in particular, study safety and efficacy of potential therapeutic strategies before moving them into clinical trials. So basically, patients can reveal the disease, but animal models are very helpful to understand the biology behind it. And this brings me to the second very important question, which is, what makes a good animal model? Well, there are many different features that can define a good model. I tried to summarize all of them in this table. The first one is, for sure, genetic validity, because you want that the animal model basically recapitulates the genetic condition, the causal defect of the human disorder. And this is something that we call construct validity, but this is also probably the easiest condition to achieve, because when we design an animal model, we have this rationale in mind, right? But this is not sufficient, because the genetic alteration that we introduce in the model should also be able to recapitulate the biochemical and molecular alterations that are typical of the disease. In the case of CTD, for example, the alteration of brain creatine levels. But not only that, also the key aspect of the human disorders, for example, intellectual disability, autistic-like features, and epilepsy. And this is what we call face validity. And this is something that we cannot take for granted, because there are multiple examples in other neurodevelopmental disorders, such as Rett syndrome or Fragile X, for example, in which there are animal models that can capture some of the critical phenotypes of the disorder, but they completely fail to recapitulate the full spectrum of the same disorder. So basically, over the years, many different animal models have been generated for CTD, and here I try to summarize with these stars how they are positioned in the literature for every single aspect that we have just discussed so far. And as you can see, we can tell that we have very good animal models for CTD because the genetic validity is pretty high, but also the face validity, and we are. Well, there are many positive results emerging in different research lines. So this is the CTD animal model landscape. I will describe all of them in the next slides, but I would like that you keep in mind that each of these models have been designed to answer a different question. So probably there is no an ideal model, but altogether they can provide a comprehensive view of CTD pathophysiology. Here I would like only to mention these models, the reporter models that are emerging right now in the literature. Actually, we have generated one of them. I don't have results to show you, but these models are made with a reporter protein, such as GFP, linked to SLC6A8, to the gene of creatine transporter. And this will allow us to understand better when and where the creatine transporter protein is expressed in the brain, but also peripheral tissues, since the lack of available antibodies that can do the job. So let me start from describing the first animal model that have been generated for CTD, and basically, I'm sorry, but the animations do not work, but whatever. Okay, this was the first animal model generated in the field and basically laid the ground for the all subsequent works. Has been generated in 2011 by the group of Matthew Skelton and carries the deletion of the exons 2, 3, and 4 of the SLC6A8 gene. And this leads to the complete loss of function of the creatine transporter protein, and importantly, this animal model recapitulates the main hallmarks of the CTD pathology, including brain depletion of brain creatine levels, alterations in energy metabolism, and cognitive and memory deficits in the Morris-Water maze, novel object recognition task, and fear conditioning. So basically, this represented a very important model because allowed us to understand that CTD can be actually modeled in rodents, and that the animal models can recapitulate the main feature of the human disease. So building on this no animation at all. Okay, doesn't matter. Building on this pioneering work by the group of Matthew Skelton, our group and others expanded the characterization of these animal models, these knockout models. And in particular, we have generated a second mouse model carrying the deletion of a different set of exons in SLC6A8, the exons 5, 6, and 7. And in addition to the brain creatine depletion and the behavioral and the learning and memory deficits already observed in the model of Matthew Skelton, we have also observed autistic-like features in these knockout mice, in particular with increased stereotype movements in the rotarot task and excessive self-grooming. Moreover, we also went beyond the behavioral characterization, and we used EEG recordings, in particular video EEG recordings in knockout mice, and we found that knockout mice display an epileptic phenotype, with these mice showing at least one spontaneous seizure in a time frame of 24 hours. And this is very important because there are not many animal models recapitulating epilepsy in modeling neurodevelopmental disorders. Moreover, we have also established a set of translational biomarkers that can be easily measured across different species, indeed using, again, EEG, but also intrinsic optical imaging and functional MRI. We have found alterations in the oscillations of EEG, in particular theta, alpha, and gamma oscillations, we found an abnormal amplitude in the visually evoked hemodynamic responses, and you will see the translation of these results with the FNIR studies that we are conducting together, Michela Fagiolini, but also with Aurore Curie at the Hospital Civil of Lyon, tomorrow in the presentation of Israel Adamatsu. And we also found widespread and robust hypoconnectivity in the brain of knockout animals, both at the level of local connections, but also in the longer range connections. So this second mouse model and this further characterization of the model allowed us to further increase the translational relevance of knockout mice in the field of CTD research, basically transforming them from a simple behavioral model to a model in which, to a very robust platform for biomarker discovery and therapeutic development. Here I would like to acknowledge that actually these knockout models do not recapitulate completely the human phenotype, because in collaboration with the group of Anna Raffaello at the University of Padua and Marco Linari at the University of Florence, we have also expanded the characterization of these mice beyond the central nervous system, and we have demonstrated that they have a striking pathological phenotype at the level of the skeletal muscle. Indeed, we found the knockout mice muscular atrophy at the structural level, a functional impairment, including reduced grip strength and reduced capacity to generate an isometric force. And this was together with profound alterations in the structure and function of mitochondria, suggesting that mitochondrial dysfunction might be one of the major drivers of muscle pathology. So this demonstrates that probably no ideal, no perfect model exists, because as we know, the muscle involvement is relatively uncommon in CTD patients. And it is important that we take this into account while we are doing preclinical studies in these mice, because probably this muscle involvement is amplified in knockout mice compared to what we observed in patients. So this is, okay, so we have described the phenotype of knockout mice. By describing the phenotype is just the first step, because we can do more with animal models, and for example, we can understand the mechanism of CTD, underlying CTD pathophysiology. We can understand how the creatine loss leads to neuronal dysfunction, circuit alteration, and at the end to the disease, actually. And this is why we use our knockout models to study the cellular alterations that are due to the creatine loss in the brain of these mice. And we have found many convergent pathways. The first one is a loss of inhibitory synapses. Indeed, we found in a strong decrease in the density of GABAergic inhibitory synapses in the brain of knockout mice, while glutamatergic synapses were largely preserved. This is not surprising, because GABAergic interneurons in the brain are high energy demanding cells, but this also suggests that there is probably an imbalance between excitation inhibition in the brain of these animals that might explain not only network excitability, because less inhibition means hyperexcitability of the circuits and epilepsy, but also behavioral abnormalities. And we will see that in more detail later during this presentation. We also found an important state of microglial activation, suggesting that there is a chronic inflammatory state in the brain of knockout mice. And finally, we found impaired hippocampal plasticity in terms of reduced neurogenesis. So we found a reduced number of proliferating cells, of newly proliferating cells, but also reduced levels of neuronal differentiation. So basically, taken together, these findings suggest that chronic creatine deficiency does not affect a single pathway, but it triggers a cascade of interconnected pathological events that involves synaptic dysfunction, neuroinflammation, and impaired brain plasticity. But this is just the first step of this work, because we have characterized these deficits in the animal model, but we still do not know how these are linked to the loss of creatine in the brain. Probably mitochondrial dysfunction might be one of the answers, but it is likely not the only answer. So much work is still needed to better understand the mechanism, the causative mechanism of the disease. Another important question in the field of CTD pathophysiology is whether creatine is fundamental throughout life, or there are critical windows during development of the brain during which creatine plays a crucial role and during which its loss causes irreversible pathological events that leads to long-term consequences. And to answer this question, there is a very elegant studies by the Matthew Skelton group using a temporally induced conditional mouse model. Basically, they found that when they delete SLC6A8 very early in development, at postnatal day 5, they found a strong and robust spatial learning deficits in the Morris-Water maze, but also in other cognitive tests. While when they delete the same gene, of course, in adult animals, they do not find any cognitive impairment. So this suggests that creatine is fundamental during brain development, but the adult brain circuits are much less vulnerable to its loss. But the developmental vulnerability is just a part of the story, because another important question is, creatine transporter deficiency is a static condition or a disease that evolves over time? Well, our answer in animal models came from our studies, longitudinal studies in the knockout mouse. And what we have found is a progressive worsening of the cognitive symptoms in these mice. This is at least partially consistent with what has been reported by the recent natural history study Vigilant. And so we think that early creatine deficiency basically disrupts brain development, but also initiates a progressive cascade of events that leads to the progressive deterioration of symptoms, at least of cognitive symptoms. So this is just a summary of this first part of the presentation. For the sake of time, I won't go into details of every single asset that we already touched, but I would like to stress the importance of animal models in CTD research. I would like to stress that for CTD, we have animal models with very high face validity, and these models have not only improved our understanding of CTD biology, but basically can establish an experimental foundation to build therapeutic development. And with the next slide, I would like to show you just some few representative examples. They are not probably exhaustive, but they demonstrate how huge can be the impact of animal models in CTD research. The first, at least partially successful story is the cyclocreatine story, because although it has been, well, the clinical development of this molecule has been discontinued due to safety concerns, we and others have demonstrated that the administration of this molecule can reverse many of the CTD symptoms. So these studies brought a very important message, which is CTD can be tractable. You can use a pharmacological treatment to improve CTD symptoms. More recently, Dodicil creatine ester emerged as a next generation derivative of creatine, and the group of Alois Mabonzo beautifully demonstrated that the intranasal administration of this molecule leads to brain creatine restoration and to the improvement of deficits in learning and memory, thus supporting the translation of this molecule towards clinical studies, thanks to the involvement of Ceres Therapeutics, and hopefully this molecule will be ready for patient treatment very soon. At the same time, you know that our group, but many other groups in the room, are working on gene therapies approaches, with the aim to restore the expression of SLC6A8 in the brain. So targeting not only the alteration of creatine levels in the brain, but the root causes of the disease. We have already demonstrated at the proof of concept level the feasibility of gene therapy in CTD. Because we have seen that can improve many different symptoms in animal models at least, but we still believe that we need some more optimization of the vector design in order to maximize safety and efficacy before moving to clinical trials. So we need to study a little bit more this approach. But in any case, I think that these studies demonstrated that the neurological phenotype of CTD is tractable, and this is perhaps the most important contribution of CTD animal models, because they transform therapeutic development from a theoretical perspective to an experimental reality, which is very important for the CTD community. Well, okay. So far, I have presented you just studies on knockout mice. And they have been extraordinarily informative, as you have seen. But knockout mice, as we have seen, carries the deletion of a portion of the SLC6A8 gene, while most patients harbor just specific mutation of this gene. So one question that might arise would be, how confident can we be that the phenotypes and the therapeutic responses that we found in these knockout models can actually reflect what happens in humans? And to address this question, the group of Olivier Bressan developed the first knock-in model of CTD. In this case, it was a rat model, and this model does not carry a deletion of a portion of SLC6A gene, but a specific mutation that was previously identified in patients. So in this case, the genetic locus is largely preserved, and the construct validity of this model is higher. Importantly, these models displayed the main hallmarks of CTD that we have previously observed in knockout mice, in particular brain creatine deficiency and behavioral abnormalities that are consistent with the CTD phenotype. So basically, we can tell that knock-in rats didn't change the story of CTD, but they confirmed that we were telling the right story. And of course, they also provide an additional experimental platform for therapeutic development with even higher construct validity. So far, we have presented just data on male models, but we know that CTD is not an exclusively male disorder. We know from clinical studies that there are females that can develop significant symptoms, sometimes comparable to those of affected males. And this is due to the random inactivation pattern of the X chromosome in the brain of humans. So to try to model this, many different groups have investigated the phenotype of heterozygous females, and they found moderate depletion of brain creatine levels and mild to moderate deficits at the behavioral level with hyperactivity and only mild deficits in learning and memory, while other phenotypes were completely normal. And similar findings were found in the knock-in rats. So none of these models were able to fully reproduce the spectrum of CTD symptoms that can be observable in CTD females. And this is why I think that we need more research on this part of the CTD research, because it would be important to have a model for females as well. So let me now move from whole body knockouts to brain-specific models. In these animals, the deletion of SLC6A8 was restricted to the brain tissue while leaving unaffected the peripheral tissues. And as expected, we found the depletion of creatine levels only in the brain, while in the heart, muscle, and other peripheral tissues, creatine levels were completely normal. Yet, despite the lack of peripheral abnormalities, these animals still develop cognitive symptoms comparable to what we have observed in knockout animals. And we also found a progressive decline of their cognitive performance, as we have seen in knockout animals, thus suggesting that brain creatine is, the lack of brain creatine is sufficient for the determinism of the neurological function, phenotype of CTD, at least for the cognitive part. Because interestingly, other non-cognitive dysfunctions, such as excessive grooming or other autistic-like behaviors, are completely absent in these brain-specific mice, thus indicating that the combination between the lack of brain creatine and the lack of creatine in peripheral tissues contributes to the expression of the full CTD pathology, while brain creatine deficiency is sufficient for cognitive impairment. In parallel with that, we also developed a plethora of cell-specific models in order to understand in the brain which are the cell population more vulnerable to the creatine depletion and which are the cellular population more involved in CTD pathogenesis. The first clue came again from a conditional mouse model developed by Matthew Skelton, in which the lack of SLC6A8 was restricted to GABAergic inhibitory neurons. And they found a key phenotype, a deficit in contextual and cue fear memory and learning. So this teaches us that GABAergic dysfunction is sufficient to cause the impairment of learning and memory, and probably this means that GABAergic interneurons are very important in the CTD pathophysiology. We then move a little bit further in this research, because we have generated a mouse model in which SLC6A8 was deleted only in parvalbuminergic interneurons, which is a subset of GABAergic interneurons with very high energy needs because of their fast spiking features. And what we have found is that this conditional mouse model shows many key phenotypes of CTD, including cognitive alterations, impaired cortical processing, and also alterations at the level of EEG, including higher vulnerability to epilepsy. So this suggests that parvalbuminergic interneurons might be one of the main contributors of CTD pathophysiology and also an interesting target for future therapeutic development. In contrast, a completely different picture emerges from the study of mice in which SLC6A8 was deleted in dopaminergic neurons. In this case, no cognitive dysfunction was observed, but only a very robust hyperactive phenotype, thus suggesting that the lack of creatine in this specific cell population might explain the ADHD symptoms that are frequently present in CTD patients. And finally, again, Matthew Skelton studied also the serotoninergic system in knockout mice, in whole body knockout mice in this case, showing a deficit in serotonin synthesis and turnover, suggesting that also the monoaminergic system is involved somehow in the CTD pathophysiology. So the key take-home message is that CTD is not a uniform neuronal disorder, but different symptoms can emerge from the dysfunction of distinct neuronal population. And all of them might be very interesting entry therapeutic points, but some additional studies are needed to better understand this part. Let me finish my presentation just mentioning one additional model of CTD that can implement animal models. Patient-derived brain organoids could be an additional human platform, very interesting for studying CTD pathophysiology, but also therapeutic development. Even if this technology is still under development, at least for CTD, they have shown already important findings, such as reduced creatine uptake and alteration in neurodevelopment. So deficits in neuronal proliferation, neuronal differentiation, synaptic protein, and mitochondrial function. All these findings are very similar to what we have described in knockout mice, thus indicating that the very same results emerged from two different experimental platforms, and so somehow validating both of them. For now, brain organoids are not ready yet to substitute animal models, and I'm not sure that if they will ever be, but for sure they represent an additional, an important addition to our experimental toolbox, because studying together brain organoids and animal models can provide complementary insights in CTD pathophysiology and therapeutic development. And this brings me to the end of my presentation. These are just few take-home messages. Again, I won't go into details, but what I want to stress is that I think that we are entering in a new era for CTD research, and this is probably, at least in part, due to the importance of animal models, because for many years we have thought to CTD as an untreatable disorder, but thanks to the work of, well, many of you in this room, CTD, and also thanks to the support of ACD community, of course, it is becoming a disease that we can treat, and hopefully we will have some treatment available for patients very soon. Thanks for your attention.
Thanks, Laura. We have time for a couple questions, okay?
Thank you, very nice talk. I had a question about the studies that suggest there's a window with the P5 and P60. I guess it's dependent on the fact that there was no findings in the P60. I just wanted to ask whether or not in those studies they did confirm that the tamoxifen was doing the same amount of pre-excision as in the younger, and whether or not, like you were showing, there's differences in inner neurons. Did they actually hit the same neurons, or is it possible that at P60 the effect of tamoxifen wasn't complete?
To be honest, I don't think that in that study there was this specific control, but I totally agree with you that this would be very helpful. Also because in our hands, for example, in brain-specific mouse models, we have found a beginning of the phenotype in adult mice. So, well, maybe further studies are needed to better understand this part, but for sure, creatine is fundamental during early brain development. This is for sure.
Hi, I have a 6-year-old with CTD, so when I hear about something like that, it concerns me because, you know, he's growing faster than the drug development process, right? So did you not see any difference in the adult mice once the brains developed? That's one of my questions. And then for the knock-in mice, because you know, even if you have the same mutation, you see different phenotypes in people. Like, did you see any different phenotypes in those knock-in mice? And then, you know, there are so many mutations. How did you decide to use, like, what kind of mutation for those knock-in models?
For the second question, this was just, let's say, an example. You can, at least in theory, generate as many knock-in mice as mutation you have in patients, because the idea of generating knock-in mice was also to direct the therapeutic development towards a more precise, personalized medicine. So this was, I think that more recently another knock-in mouse model has been generated with the different mutation. I don't remember the details, but at least in theory, you might generate a mouse model for each different mutation. I don't remember the first question though, sorry. Well, this is something that we have to study for sure. And one of the most important things to be studied in therapeutic research is what happens if you give creatine supplementations or other molecules or even gene therapy, not only during a very early time window of development, but also in adults, for example. And this is one of our aim in our research. For example, for gene therapy, for now, we have demonstrated feasibility giving the viral vector very early, but we would like also to test what happens if you give the same treatment in, let's say, adolescents. So it's important to cover the whole lifespan to see what happens for therapies.
I'm just going to add my standard encouragement for CTD parents that we have adults here diagnosed with GAMT, and their family is very happy to see the progress that they are making now that they have creatine. So differences with length of time, of course, yes, but all hope lost, absolutely not.
Thank you for the talk. That perfectly sets the stage for our next one. We're going from big to small. Our next talk will be delivered by Aleksander Bogoniewski. Alex is a fourth-year PhD candidate at UCLA. He studies the neurodevelopmental consequences of SLC6A8 and GAMT deficiencies using humanized model systems, specifically iPSCs and iPSC-derived organoids. He is evaluating the efficacy of current treatment regimens on neurogenesis as well as neuronal function, specifically exploring AAV-mediated gene therapy. Alex, over to you.
Thank you, Sangeetha, for the warm welcome. Today I have the distinct pleasure of sharing with all of you what we have been finding out in the lab, which I have titled Growing Human Stem Cells and Brain Cells in a Dish to Better Understand and Treat GAMT Deficiency. Before I get into the science of the talk, I wanted to give a quick background of myself. I come from a very large family. I'm the oldest of 8 siblings, 3 of which have been diagnosed with autism spectrum disorder, and that's me. My sister is blocked by my brother on the scooter. Here's us again in 2022. Again, sister not there. And one of the last times we took a family picture all together was in 2020. And again, there's me there. But I want to talk specifically about my youngest brother, whose name is Ariel. And he was diagnosed with autism spectrum disorder at 1 year of age. And mostly due to familial history of having this disorder, he underwent a full genetic sequencing panel. And it identified a mutation in a gene called TAB2. And it's a very rare mutation that's not identified in any state's newborn screening panel. And it's associated with defects in connective tissue and heart function as well. He also has a shorter stature, and it also impacts his intellectual development as well. And he has been nonverbal. He's currently 6, but he was nonverbal until around 4.5, 5 years old. And we previously communicated primarily with American Sign Language, and there was a lot of babbling and not full sentences until around 5 years old. Fortunately, he just finished his first year of kindergarten, and through different types of therapies, which I have helped to administer, different types of speech therapy, physical therapy, and Jasper-style play therapies, he seems to be at an identical development level to the pediatric standards. And Ariel loves Godzilla, and he also loves the LA Zoo. So this is where we go on a lot of weekends, and this is where he is in front of the otter exhibit. So now I just want to introduce GAMT deficiency, which Dr. Longo did a great job of introducing earlier, which is characterized by a lack of creatine production. And this leads to a lack of intracellular creatine within the brain, musculoskeletal system, and the heart. There's also a large accumulation of guanidinoacetate, acetate, which has been shown to bind to GABA receptors and lower the threshold for seizures. The current treatment methodology involves a supplement regimen of creatine monohydrate, L-ornithine, and sodium benzoate, and the main goal is to increase the amount of creatine to overcome the bioenergetics of the disorder and decrease the activity of AGAT by also limiting arginine through a protein-restricted diet. Now, these treatments have provided benefits. However, intellectual disabilities do appear to be non-reversible based on when the treatment has started. And for a lot of the science that we're working on, it involves this central dogma of biology, where DNA is turned into RNA through a process called transcription, and then it's turned into protein through a process called translation. And a lot of the work that I have been looking at has involved RNA and protein. And so, for example, when there's a mutation within the DNA, that leads to an RNA that would have that same identical mutation as well, which would lead to a misfolded or a non-functional protein, which would be the present, for example, in GAMT deficiency. And my research primarily involves using human models. So I use stem cells and organoids, and we'll get into that in a minute. But the primary method of studying CCDS has been and continued to be mouse models, which have been extraordinary at understanding the biochemistry of the disorder, and they provide a great platform for evaluating different treatments. But there are some differences between mice and human that are important to understand before we move forward. Starting off, the size of the brains are quite different, where the human brain is quite larger. When you account for weight based on body size, it's around twice the size. There's also differences in the prefrontal cortex region, where the human brain has a much more developed prefrontal cortex. There's also some regions in the mouse brain that are not reflected in the human brain and vice versa. And in order to fully understand CCDS, we should be using human model systems to supplement our ongoing mouse data. Then using a resource at UCLA, it's called BrainSeq, which what they did is they took RNA, so again, that's the reads of the DNA, and they analyzed how much expression is present within mouse and brain tissue, and human brain tissue. And what they found, what I found through analyzing the data, is that GAMT specifically is expressed around 25 to 1,000 times higher in the mouse brain than the human brain. And this pattern holds true with SLC6A8 expression as well. However, AGAT expression appears to be relatively similar, except in endothelial cells, which make up the blood-brain barrier. All of this continues to prove that we should be using human models to supplement our current mouse data. And the human model system that I chose were induced pluripotent stem cells, which are generated by reprogramming a patient-derived tissue. So for example, I have a flow chart on the diagram here, and I'm going to use the screen on the right-hand side as it's closer to me. But through the Coriell repository, there's been either blood samples or skin samples that were harvested from patients. And what they do is they isolate these types of cells called T cells from the blood. What they then do is they add these 4 reprogramming factors here that allow them to enter a more stem-like state. So over a series of media changes and a series of, they call it passaging, you're selecting the cells that you would like, you end up with these cells that are called stem cells. And what's special about these stem cells is we can turn them into any single type of tissue except for sex cells and except for the placenta. They are induced pluripotent stem cells. And what we've been doing in the lab is we've been taking these stem cells and turning them into miniature brains and neurons as well. So for example here, we can turn these iPSCs into neuron cells to better understand and treat CCDS. We can also use this approach. It's called CRISPR-Cas9. It's had a lot of media attention on it recently. What it is, it's an RNA that binds to a protein that acts as a pair of molecular scissors. So you can actively cut out or you can replace certain genes, which is what we've been doing in the lab. One of the cell lines that we were able to successfully generate involved a CRISPR-Cas9 cut where we knock out GAMT from a normal wild type line. And this is an incredible technique because we can isolate the effect of the specific mutation instead of comparing a patient line to a non-related wild type line. And through the neuroscience that my project involves, we start out with these stem cells, which are present up top. And these are the immature cells. And as these cells mature, they turn into neuronal progenitor cells. These cells are able to be differentiated, meaning turning into the 3 main brain cells, including oligodendrocytes, neurons, excitatory and inhibitory, but my project is excitatory, and then astrocytes as well. So in summary, we can take a blood or skin sample, we can reprogram them into induced pluripotent stem cells, and my project involves making miniature brains and involves plating brain cells on a dish. The miniature brains look like this. the neurons look more similar to this. And the huge advantage of using these organoids is that all 3 brain cells are present within the same system, so we can evaluate neurodevelopment and we can evaluate different delays that are present within the system. And with the individual brain cells, they act as a very rapid source of treatment, where the cell line that we're using, we can get mature neurons within 12 days. And this involves only human samples, there's no risk to patients, and it involves a rapid testing of different therapeutics. So starting off with our organoids, we were able to make organoids from GAMT patient lines. Both of these were from the Coriell repository, and we used 2 wild-type controls labeled FiberGrow and XCHuff1, and we were able to grow them for the full time course. We did 100 days, and at day 50, here's 49, we found that they did not grow differently compared to each other. So they show a relatively similar rate of proliferation. What we then did is we took these organoids and we cut them into very small segments, and we analyzed for different levels of protein. So on the top of this image are GAMT organoids, and on the bottom are FiberGrow, our wild type control. And starting with the left-hand panel, which would be this panel here, there were several organoids, then this trend holds true, and the amount of yellow is MAP2. That is a mature neuronal marker. We can see that the bottom one, the FiberGrow, appears to be more bright, and it appears to be more red as well, showing overall more neurons. In the middle panel is PAC6 in red. These are our neuronal progenitor cells. We don't see much of a difference. And then on the right-hand panel here are Ki67 in green and GAMT in red. So we see GAMT staining with the organoid, and we also see Ki67 staining with this organoid as well. We then took these organoids, and we had a total of 9 across 3 samples, and we did something called RT-qPCR, which analyzes the total amount of RNA within the spheres. And what we found is that the day 50 organoids have fewer post-mitotic mature neurons, as indicated by NuN expression on the left-hand side, and there's no difference in PAC6 neuronal progenitor cells. So it's not that they're proliferating or at a different rate, but they're differentiating at a different rate. Now, this trend was further exacerbated at day 100, where we stained for oligodendrocytes in green, astrocytes in red, and mature neurons in yellow. So this staining here represents the full 3 brain cells. And when we analyze the spheres, what we find is that these day 100 organoids have fewer mature neurons, as indicated by synaptophysin. They have fewer oligodendrocytes by OLIG2, and they have fewer astrocytes by GFAP. We then did the same thing that we did before, where we did RT-qPCR, so looking at that RNA level, and we found that these organoids have elevated neuronal progenitor cells, the GAMT organoids. So going later into development, they're still having these more naive-like cells. They have no difference in NuN, which are post-mitotic neurons. However, a huge finding that we had was that the amount of CTIP2 positive neurons, which are a deep layer 5, layer 6 neuronal marker, is significantly delayed within the GAMT organoids. So we're showing all 3 brain cells have been delayed within these spheres. However, with these organoids, we are comparing a GAMT patient line to a wild type control. We went back and we edited a line of induced pluripotent stem cells, and we did a knockout of GAMT. So on the top of this slide here, and it's incredibly difficult to see, but these are 2 DNA strands, and our knockout is indicated by this red region here, where it's completely blank. So we have a successful CRISPR-Cas9 mediated knockout of GAMT exon 1, which impacts the amount of GAMT that can be expressed. We then did immunohistochemistry, which is staining for proteins. It's very difficult to see, but the most important part is that in the isogenic control, meaning this is the line that's before edited, there is an abundance of GAMT expression, and then in the GAMT knockout, there's absolutely nothing. Then, through a collaboration with Dr. Inga Golia at the ARUP laboratories, we then performed mass spectrometry on these lines. And what we found is that guanidino acetate is significantly elevated with the GAMT knockout. Creatine is significantly decreased with the knockout, so much so that I have to break the graph in half in order to properly demonstrate where the creatine in the knockout is. And creatinine was also reduced in these cells, which does recapitulate the phenotype that's seen both in patients and in mice. We then evaluated creatine supplementation on these cells, and we did a growth rate on a machine called CellExpress AI. So this machine, I coded a machine learning AI program, and it's grown on a machine that automatically images cells at a specific amount of time. Here is 24 hours. And what it does is the AI traces the edge of the stem cells and it evaluates the growth rate. And this same program can be applied for neurons and organoids as well. And what we found is that creatine supplementation has no impact on cellular growth rate. And not only that, through a series of 3 runs, this is one of the 3, there is no difference in growth rate of these stem cells between GAMT and the correct in the isogenic control line. We then did RT-qPCR, so again analyzing the RNA level within these cells, and we found that both AGAT and SLC6A8 were significantly reduced in these knockout cells, confirming a disruption of the creatine pathway. Not only that, SLC6A8 is around 10% or less of the regular expression of the isogenic control. We then evaluated creatine supplementation on these stem cells, and what we found is that after 24 hours of supplementation for AGAT, there was no difference in the amount of AGAT expression. There is one outlier that does bring the data down, but we can contribute this to sequencing noise. We then evaluated SLC6A8, and we see the exact same thing. So 24 hours of creatine treatment had no impact on the amount that these genes were expressed. We then left the creatine within the cells for a total of 72 hours, and we did this RT-qPCR again. And what we found is that there's minimal increase in AGAT. AGAT appears to be between the middle of the knockout and the isogenic control, but we need to do RNA sequencing to further confirm this finding. But what we did find that is explicitly clear is that SLC6A8 expression increases to around 75% of the normal isogenic control. So there's some metabolic reprogramming happening within these cells somewhere between 24 and 72 hours that allows SLC6A8 expression to rebound almost completely to normal. So in conclusion, our GAMT organoids are maturing slower. They have a fewer abundance of mature neurons, oligodendrocytes, and astrocytes, the 3 main brain cells. Then our GAMT knockout iPSCs model the biochemical phenotype of the patients and the mice, as indicated by the increased guanidino acetate, decreased creatine, and creatinine by mass spectrometry. There's also an absent GAMT protein by the immunocytochemistry. They also demonstrated reductions in AGAT and a really significant reduction in SLC6A8. Creatine supplementation partially rescues gene expression, but particularly rescues SLC6A8 expression. And it has no impact on growth rate. And lastly, these human model systems provide a platform for testing future therapeutic strategies. And that leads us into my future directions. Right now, I currently have neurons that are growing on the CellExpress AI system. And what's special about this system is that the media is being changed while I'm probably presenting at this conference. So it's very simple. There's no operator variability where, for example, if I were to take the cells out of the incubator, it would be around 30 minutes of total time. This system is able to cut that in half. So it's really, the data is very clean. We're evaluating a neuron screen of the GAMT knockout and isogenic controls right now. We're also evaluating 50 micromolar and 500 micromolar of creatine monohydrate supplementation on these neurons. We're also evaluating 5 micromolar and 30 micromolar of guanidino acetate on these neurons. As with GAMT deficiency, we're not sure if it's a combination of the absence of creatine and abundance of GAA, or maybe it's due to primarily GAA accumulation. We're going to be taking these neurons and we're going to be doing something called RNA sequencing on them to get the full profile. And through this profile, we'll likely find out what changes at around that 72-hour time point of creatine supplementation. We are also adding a GAMT cDNA. So this is the coding DNA, so only the exons. We're going to be adding it with an antibiotic-induced promoter. So we can get anywhere between as low as 25% to over 100% GAMT expression within these neurons. And we're also going to be evaluating an AAV9 gene therapy carrying a very similar GAMT cDNA. And what we can do is we can plot the AAV on the total percentage of GAMT expression to get a correction efficiency of the virus. Now, this work did not happen alone. I just give a huge shout out to both Dr. Lipschitz, Dr. Reyes, and Susana Rivera, who've helped with this project. Of course, Dr. Inga Golia, who without this help, we wouldn't have the mass spectrometry data, the molecular screening resource, ACD, for continuing to fund this project. And I am especially grateful that I have both Dr. Longo and Dr. Lipschitz on my dissertation committee, and they have contributed so much intellectually to the development of this project. So I'll stop now and take any questions that you have.
I have a question. I see that Laura's not in the room, but maybe there are others who can chime in. So to the point that you made about seeing reduced expression of GAMT and SLC6A8 in the engineered cell lines, I was wondering if anyone has studied this in the mouse models, and do you see anything similar? Is there anyone that might be able to address this? Do you see changes in expression of SLC6A8 or AGAT in the CTD mouse models?
Congratulations, really excellent results, but they are also stunning and I have no explanation, and I will talk to you later about that. We haven't studied CDD mouse, but we are hopefully now finally publishing our mouse study on GAMT deficient mice that we treated with creatine for 10 weeks. And we assessed effect of creatine treatment, of course, on growth, but specifically we were looking for the effect of creatine treatment on guanidino acetate and why does it lower guanidino acetate? Because all GAMT patients, and Nicola didn't mention that, when they get creatine treatment, it's not only to replace the creatine in the brain, it is also to inhibit AGAT expression on a genetic level, and that lowers guanidino acetate by 50%. That's what we know from patients. And we wanted to study this in the mouse to better understand how is this mechanism. And to say it short, we have evidence that creatine treatment reduces guanidino acetate in brain, muscle, heart and skeletal muscle, but not in the liver. And it is by reducing the AGAT expression, both on the mRNA level and the protein level and the activity level in all those organs, brain, heart, muscle, kidney, but not liver. And the effect of creatine on downregulating AGAT is very strong. We had this before in many other cell lines, and so this is why I said it's stunning to see the opposite when you treat with creatine.
And then I want to add something that for the data specifically for, it's going to take a while to skip back, but for SLC6A8 and AGAT, I also tested the two GAMT patient lines from Coriell, the iPSCs, and they showed pretty much identical phenotypes, so identical levels of expression. So we're seeing some sort of rescue biochemically with creatine at that 72-hour time point.
Thank you. Just thinking from a drug development perspective, whatever a small molecule drug development perspective, I just wanted to get your opinion on how, or the state of the art in terms of looking for a pharmacodynamic biomarker in any of these models, and if you expect any of the mouse models to be relevant to humans in terms of biomarker, because there is quite even treating with creatine, right? There's quite a dose range variability depending on if it's GAMT or AGAT, but even within that specific indication or cohort. So I'm just wondering if there are any reliable biomarkers or research on reliable biomarkers that could help folks who are developing drugs, test them in these models. Yep, thank you.
Right. Great question. So with the neuron model specifically that I'm generating, it allows us to open up the door for evaluating many different therapies on that model. Through the RNA sequencing that we're doing as well, we can also, if there's any hits, for example, that genes that are elevated or decreased, we can evaluate those further as well. I'm not an expert on the mouse models, as most of my work focuses on these 2D models, but I think the validity of taking the mouse treatment and evaluating them in humans or vice versa is going to be what's going to accelerate the therapeutics.
So I think the question was whether anyone's working on transition state inhibitors or the like, particularly given what we know about feedback regulation of SLC6A8 and as well as GAMT and AGAT. Maybe this is a conversation that we can continue over the lunch. For now, I would like to thank Alex for his wonderful talk. It's my pleasure to welcome Dr. Emil Alexov to our conference as well as this next talk. Dr. Alexov is a professor of biophysics and bioinformatics at Clemson University. His work focuses on understanding the molecular mechanisms underlying the biological processes, in particular in silico drug discovery as well as protein-protein interactions. Dr. Alexov, thank you. Just note that there will be children coming in, so I apologize.
I'm just going to clarify in case anyone's confused. If your child went to the NAC, the National Ability Center, you do not need to get up and get them. They're having lunch over there. Only if your child did not go.
Thank you for the introduction. Thank you for the introduction, and thank you for the invitation to give a talk. Before starting my presentation, I simply like to speak a little bit about our research as a general topic, and then to make a small comment. As it was said, our lab is computational biophysics lab. We use wave physics to understand how protein, DNA, RNA work, and we use computers. We don't do experiments. And our goal is to understand how they work, these small molecules, protein, DNA, RNA, and to infer how genetic variants alter this function, and when we understand that, to develop drug that will partially or completely eliminate the disease-causing effect. Second thing that I like to say before my presentation is that I'm taking point of Heidi introduction. Amount of parents and patients is larger than scientists, so I'll be changing my presentation to a way to be understandable from non-specialists. You'll see a lot of charts in my presentation, but I will go very quickly over them and trying to verbally explain the results rather than making you thinking science. And scientists do not criticize me for that. I don't know how to advance the slides. Doesn't. No, it works. So I'm gonna skip this slide. This was nicely explained by previous speakers. I even gonna skip this one, except that on the very right side of my slide, I simply underline the two basic mechanisms for creatine deficiency syndrome. Over here is the panel that indicates the defect in the biosynthesis, which seems to me is not so critical. It could be partially treated with creatine supplements. On the left-hand side is the topic of my presentation now, namely how the defects of creatine transporter cause the disease. Being trained as a physicist, when I need to address questions about disease treatment, my first step is to understand why it does not work, why the creatine transporter does not work when there are mutations. And in order to address that, I need to understand how the creatine transporter works. So this is going to be my major topic of today's lecture. Also, I'm going to skip this slide because it is a repetition of the previous slides, previous speakers. And now the topic of my presentation today. This slide attempts to schematically illustrate the computational approach of modeling biological macromolecule. In cartoonish presentation, the creatine transporter, which is membrane protein, is represented by this purple object, and it is immersed into a protein, into a lipid membrane. This is the cell membrane. And on the top and on the bottom are small compounds, either creatine or some ions. So you understand, in order to model the transportation of creatine through a creatine transporter, we need to have a driving force. Something must be pulling this creatine through the creatine transporter. And this is so-called electrochemical gradient. I don't want to underestimate your knowledge, but this simply implies that one thing is going to move from a high concentration to a larger concentration. And there might be some other forces like electrogradient coming from the membrane potential, which again, I'm not going to give you numbers, result into a pulling force about several piconewtons. These are very small numbers, but in micro world, protein, DNA, RNA, newtons, angstroms are very large numbers. So there is no prior knowledge. There are no experimental studies to indicate what is the pulling force in vivo. So then we've been facing this challenge, and we need to do some kind of estimation of what the pulling force is, which I'll explain in a couple of slides. Before getting to this topic, for people that are more in molecular level of explaining the details, it is known that the transportation of creatine through the creatine transporter goes to 4 different stages. I'm not going to read this thing for you, they're clearly marked, but essentially, as the creatine goes to the creatine transporter, the creatine transporter must change once, 2, 3, 4 times, and to pull the creatine out inside the cell. What is known so far? There were quite many experimental studies attempting to figure out what are these conformation changes, and they were successful for 2 of them, marked here with this triangle, namely the stage when the creatine is already almost ready to get out, and when it is going to be released, there are crystallographical structure. Again, speaking about parents of the patients, for computational modeling and some other studies, it is crucial to know what is the three-dimensional structure of the target of the protein. Once you know it, then you can ask questions of what is happening. So that is what we knew. There was also another study, computational, that make a prediction of what is the structure of the protein prior the creatine gets inside. And what is unknown is this one, when the creatine is already in the pocket. So our study will start from here and will attempt to go through all these stages one by one by the means of molecular dynamic simulation. Molecular dynamic simulation is a technique that uses principle of physics and laws of physics to follow what are the changes of the position of individual atoms inside the proteins and other molecules. So this is what it's going to do. For my fellow colleagues, if you are interested in details, the work is published already a month ago. And with this work, we illustrated how the creatine is transported through the creatine transporter. The goal of my study is to go through these different stages, namely to figure out what is the details of creatine transportation through the creatine transporter, then to figure out how the missense mutation, I'm mostly interested in missense mutations, how missense mutations affect this transport. And on the last stage, to do targeting drug design of small molecules that will bind to the affected mutant creatine transporter and partially or fully restore the transportation. Just for illustration, I'm not going to spend much time. This is our computational system. As you perhaps can appreciate, the protein is shown in green, surrounded by a lipid bilayer, several million water molecules in our system, along with sodium ions, chloride ions, and the creatine itself. And the thing that I've mentioned to you, we need to figure out what is the pulling force that drives the creatine through the creatine transporter. To make long story, and as I promised to you, I'm not going to walk you over all these graphs and complicated stories. We attempted to start with a pulling force of several piconewtons up to 100 piconewtons. What should be our criteria to decide which force is physiologically relevant? First of all, our study aims to reproduce experimentally observed things. How the creatine is released, what is the sequence of the release of sodium and creatine ions, not to completely distort the structure because you can drill this protein, so to say. And based on this consideration, we figured out that the optimal pulling force for putting the creatine inside of the pocket is 20 piconewtons. In that case, we were able to see the conformation changes that are expected, such so creatine comes inside of the pocket, the mouth of the protein is open. When the creatine comes inside, the mouth closes. And then the next step is to go through the core of the protein to be released inside of the protein. Which brings me to the other topic. Some of the conformation changes that are required for the transportation are long-lasting. Even nowadays, with all these advantages, computational power, some of these timescales are not achievable. That forces us to apply something that is called targeted molecular dynamic simulation together with a constant force steered molecular dynamic simulation. Thing is that I'm not going to discuss with you. I'll be happy to have a private conversation. And that resulted into finding another effective force that pulls creatine and sodium ions much smaller than the original one, about 5 piconewtons. One of the key criteria of identifying what is the best pulling force and how successful our modeling is was to reproduce experimental observation of how the sodium ion, creatine, and other sodium ion are released in the time. So vertically is the time, and I showed what is the observation in our molecular modeling. The first release is sodium 2, which is experimentally determined fact, then the creatine, and then the sodium 1. Not certain to what extent should be talking about such details, but just to give you an idea how important the details are in molecular modeling, I'm going to spend just a couple of minutes of a little bit more details that illustrate what was done. You can assume that simulation that involve a large protein of several 100,000 atoms, lipids, and several million water molecules is quite complicated thing. But what is the role of these million water molecules? Are they just surrounding the protein, doing nothing? The key finding of our investigation was that water molecules are essential for letting the creatine and sodium ions get outside. This is perhaps the most difficult part of my talk, to how to express or explain the thinking for people that are not in the field. So in molecular modeling, typically we report the things in time. What is happening as the time of simulation progresses? And this is my horizontal scale, the time of simulation. And vertically, we indicate the amount of thinking. So if you follow this red line, this indicates how much water molecules come into the pocket throughout the time. And the blue thinking indicates how many contacts either sodium or creatine makes with the surrounding residues. What do you expect? When the creatine moves away, it should start be surrounded by many, many water molecules and pull out, and the number of contacts should decrease, which is indicated by this green curve, and the water molecules by the red curve. They get more and more. This graph is even more difficult to explain. In molecular dynamics simulation or molecular biophysics, one important quantity is the energy. How happy a given thing is at different stage. Typically, we refer that as a free energy of state or interaction energy. So we ask the question, as the creatine is in the pocket and the protein undergo different changes, how the interaction energy change between the creatine and the protein, between sodium ions and the protein, et cetera, et cetera. And these are the energies for the first sodium ions for creatine and for the sodium ion 2. The take-home message is that we like to see that this energy gets more and more favorable, more and more negative as the creatine or the ions are prepared to leave the protein. And now the outcome of our study illustrated in easy understandable computer animation, where I show the creatine. The membrane is removed for the simplicity of the presentation. And you can follow the motion of the creatine through the creatine transporter, shown with yellow, and the sodium ions shown in blue. And you'll see that it requires a lot of jungling around. The protein needs to adopt different stages. And finally, you see now the water is coming in, red dots. The protein pocket gets soaked with water molecules, and that makes the substrate go out. So based on this study, we're able to enumerate what are important residues on the pathway of creatine that provides a knowledge to identify crucial residues and eventually what is the effect of missense mutations. I'm going to skip these things, I promise you. I'm not going to discuss the detail of this study. I don't think that they are particularly relevant for the parents and maybe not of my fellow experimentalists. So what is our goal? So far I reported published data, recently published data. Now we are working to address the question, can we identify what is the effect of pathogenic missense mutations in creatine transporter? How they affect the transportation? And then, as was asked by somebody else, can we find a particular target on the surface of the protein, such so a single drug to eliminate the effect of all pathogenic mutation? It is impossible to think that for each mutation we have a drug. Somehow we need to collapse all the genotypes into a single molecular effect. This is what we are doing so far. As it was mentioned before, we are dealing with different types of mutation, but for us, important mutations are mutations that change the amino acid sequence. Missense mutation, they can be benign, they can be pathogenic, and they can be something that is not known yet. As it was mentioned in a previous talk, the deficiency of creatine transporter can be due to two things. It cannot be transported to the membrane, or it is already transported in the membrane but does not do the creatine transport. And it was mentioned that in some cases, the defect of not be able to deliver the protein to the membrane can be rescued with some chaperones. I prefer to call these molecules not chaperones, but drugs. As a molecular biophysicist, for me, chaperone is a large protein that helps folding of other proteins, but doesn't matter. Here, simply to illustrate what is our target, we studied the literature and we found quite many experimental papers reporting what is the effect of missense mutation on either transporting the protein to the membrane or ability of the protein to transport creatine through the plasma membrane. For now, we decided not to pay attention to the missense mutation that impair the embedment of the protein in the membrane, but rather mutations that impair the creatine transport. And these are experimentally observed mutations shown to really affect the transport. And there are two controls that are known not to affect these things. If you just stare at my presentation, perhaps you will notice that here is the core, the binding protein. With red are the pathogenic mutations affecting the transport, and with blue are the benign. As to be expected, quite many of the red mutations, pathogenic mutations, are close to the core, but see, this one is far, far apart. This one is also relatively far apart. We anticipate that somehow there is a network of interactions, so-called, you name it as you want to, allosteric signaling, allosteric pathways, such so if we can identify how these allosteric pathways are interrupted by this mutation, we might be able to find a magical place where binding a small molecule will reverse the pathogenic effect. And this is what we're doing right now. No words, I simply explain our tactic and our strategy several times in this presentation. I like to acknowledge the people in my lab, especially this individual, Pitambar Podel. Most of the thinking that I described to you today was done by him, and he is currently doing in silico drug discovery to find possible drug candidates for rescuing the function of creatine transporter. He's going to graduate next year, and if you're PI in a commercial lab or something, consider hiring him. I also like to acknowledge the funding from NIH and support of Clemson supercomputer. And thank you for your attention.
Thank you so much, Dr. Alexov, I especially want to thank you for simplifying certain contexts.
I tried.
Yes, absolutely. I appreciate it. I want to pause for a minute and ask if there's any questions.
I like to say something before. I very much like to find experimental labs or people willing to work with us, experimentalists.
You are calling for it. Alex, thank you very much. I have to say, I was reading your paper this week. It took me 2 nights sitting with my pipe. I had to stop after first night because it is so tough. And I guess everyone has seen this now. But I was extremely excited about this very deep insight into the mechanistical aspects of it. And I was thinking, at the end of your paper, you say, you use those patient variants, and that is kind of something that can be now used, for our purposes, for translational research. I still don't know exactly how, but I want to, just to say that. But I have a question. You talked about the hydration of the pocket, and then this basically then allows the release of the second sodium, then the creatine, and then the first sodium. Do you have quantitative information about how much water goes into the cell during this process?
Yes, we can explicitly enumerate how many waters are entering the pocket during the release, and they are different for the first sodium than for creatine, and there are much more for the second sodium ion. I misspoke.
But the netto effect of water from the outside going into the cell during this process?
I have no idea. But you understand, the interior of the cell contains much more waters involved in the transport. 20 or 12 water molecules difference will make no change in the water contained inside of the cell. I don't think that these are linked to the water transport across the cellular membrane.
Again, thank you. I do appreciate the delving into this level of the science, because I do think it can be quite helpful. I'm just wondering, have you looked at the mutations that you're modeling and mutations that actually exist in other SLC6 family members, like the GABA and dopamine, to see if they usually cluster in a similar way, and whether it's unique to creatine, because there are deficiencies, and people who have deficiencies in those other family members which could benefit or even supplement your work.
The short answer is no. We just entered this area of creatine transporter deficiency. So far, we've been working on X-linked intellectual disabilities of soluble proteins. This is the first membrane protein that we are studying. It will take some time, but this is really direction worth to explore, for sure. No, in that case, modeling was done by somebody else, and this is homology modeling. I believe that homology modeling is better than AlphaFold when the sequence identity is very high between experimental structure and the structure that you like to model. So I'm quite confident that structural modeling is fine.
Kicking this afternoon off, I'd like to welcome Hana Young. Hana is a GAMT parent to Tilly, age 13. She's also an NHS midwife from UK. She brings her unique perspective both as a healthcare professional as well as a caregiver navigating the realities of rare disease. She's a part of the Parents 2.0 program with ACD. Hana, we're so happy to have you here with us today.
Hi everyone, can you hear me? No? Now? Hello, everyone. My name's Hana Young, and I live in Portsmouth on the south coast of England with my two children, Tilly and Arlo. Tilly was born in 2012. She was a very happy, healthy baby. All was well, and I remember celebrating when her newborn screening came back all clear. Oh, how naive I was. Time passed, and Tilly began to become further and further behind her peers. She went from failure to thrive to mild to moderate to severe global developmental delay. I knew something was wrong, but no one would take me seriously. I was told I was a neurotic first-time mum and needed to find something real to worry about. Anyway, seizures began. No medication would work. Her neurologist was adamant it was an undiagnosed degenerative neurological disorder, and genetic testing began, slowly. 1 or 2 conditions every few months. We would wait 6 to 8 weeks each time. It's not that, on to the next one. I begged and I screamed for genome sequencing, and that happened in December 2015, when Tilly was 3. And in that time, things got worse. She lost her ability to walk. I had to extend my house and moved her downstairs ready for when I couldn't carry her up the stairs anymore. Her pediatrician would talk to me about anti-suffocation pillows to limit the risk of SUDEP, and I would wake up every morning and pray she wouldn't be dead in her bed. 2 long years later, I got the phone call from Tilly's very overexcited geneticist. We have a diagnosis, guanidinoacetate methyltransferase deficiency, and it has treatment. I won the genetic lottery that day, but for us, the win came too late. Tilly has a profound learning disability. She's nonverbal and needs support with all of her care needs. She has challenging behavior, and violence is a problem. Oh. Like the other parent speakers we've heard from, I also have a professional insight. I am a midwife, and in the UK, maternity care is midwife-led. I am the one that gains the consent, and then I physically take that newborn blood spot test. How bittersweet it is to know that those 4 little spots of blood that I am so adept at taking were all I needed to save my child from a severe learning disability. I don't have much time to talk about it, but in the UK now, we are trialling offering whole genome sequencing to newborns. I take that cord blood, and GAMT and AGAT are on that test. The results come back in 2 weeks, not 2 years like Tilly's did. I cried very bittersweet tears the night after I took my first cord blood sample that would test for the condition that took so much from my child. But it's not all doom and gloom. Treatment, in our case, creatine, ornithine, and a low-protein diet for GAMT, gave me my girl back. Before treatment, she was a blank slate. The lights were on, but no one was home. She couldn't bear me anywhere near her, and she was a very angry and sad child. Life was very bleak. But treatment stopped all seizures. The lights came back on. She came back to all her strength, and she can swim and run, and she could be a Paralympian if only she'd follow instruction and go in a straight line. I always say she is joy personified, but she'll make you bleed even with a smile on her face. And then last week, she said her first word, Mum. At 13. Every year we celebrate the day she was diagnosed with cake and balloons because we know how lucky we are that her condition has treatment. Research is what you do changes lives. It changed my life, and it gave me a lifetime with my child that I didn't think her uncontrolled epilepsy would give me. I am eternally grateful. It's the first time I've done a speech without crying. Yes. Families, you're not alone. I remember reaching out to Heidi 8 years ago, and before that, I felt very alone in the world. And since being a member of the ACD, I've never felt alone again. Thank you.